Good afternoon, everyone, and welcome today to this webinar on approaches to early cancer diagnosis and the impact of artificial intelligence in oncology. Welcome, everyone. Welcome to the speaker. I'm very happy to be here to moderate this session. I'm a moderator for today. My name is Federica Zanca, and I am a program manager for medical imaging and AI in healthcare at the European Innovation Council and SMEs Executive Agents, EISMEA. And at the AEC, we support cutting-edge high-risk innovation with the potential to transform healthcare and improve patient lives. Today, we have three excellent projects, one supported by us, but the AEC, PHIRE, two others which are supported by Pillar 2, Societal Challenges, PANCAIM and PANCAID, and I'm very pleased to introduce one by one the speaker along with the project they represent. I would like to start with Dr. Massimo Alfano from the project PHIRE. Massimo, the floor is yours.
Okay, so thank you, Federica, for the introduction. I'm the coordinator of the project PHIRE. The project is about bladder cancer, which is the 10th most common cancer globally, with more than 500,000 cases every year. But it is the most expensive cancer to be treated, with more than $10 billion every year. Why is it so expensive? The reason of such a high cost is due to the presence of residual disease, which is present in the majority of patients due to two limitations. The first limitation is in the diagnostic imaging, because even during the intravesical cystoscopy, the surgeon is able to detect and to resect only the visible tumors. That means the tumors that are smaller than 1 or 2 millimeter size, they are not detected, and so they remain in the organ. And this is particularly relevant for the in situ bladder carcinoma that always appears as a flat lesion made of few neoplastic cells and always as a high grade. The second limitation is in the treatment, as the majority of the tumors, also the intravesical therapy that follows the surgical intervention fails in about 50% of the patient. As a consequence of these limits, the current diagnostic and therapeutic approaches are not sufficient to remove completely the disease. Thus, the same patient undergo multiple intervention, intravesical treatment, and routine follow-ups, with an important impact on the quality of life of the patient and on the social cost. The PHIRE consortium has designed an innovative solution for the detection and the treatment of small bladder lesions smaller than 1 millimeter. The proposed approach is based on the intravesical installation of targeted gold nanorods, which, upon irradiation with pulse light, act as a contrast agent for photoacoustic imaging, an imaging that has a lateral resolution of about 100 microns. At the same time, the gold nanorods can be irradiated with continuous light and to induce thermal ablation of the tumors. We demonstrated the feasibility of this solution in a preclinical model. In this model, the bladder cancer is detectable here on the left by ultrasound imaging about after 9 days from the time point from when we induce the tumor. And after 9 days, the tumor progresses very rapidly, and after at the time point of 22 days, it fills the entire volume of the bladder. On the other hand, here below, we also use animals in which the tumor was not detectable by ultrasound imaging, but the lesions smaller than 1 millimeter size in green were detected by gold nanorods assisted photoacoustic imaging. We then apply gold nanorod assisted hyperthermia, and 2 weeks later, we were able to detect only 1 lesion out of 11 that we detected 2 weeks before. Furthermore, the volume of this lesion was only 14 microliters. That is very low compared to the untreated animals. Overall, Overall, this solution has several advantages compared to the current solutions, because it overcomes limitation on the current diagnostic imaging, because we are able to detect very small lesions. The therapy, the gold nanoresistant hyperthermia is a thermal therapy, so it is not affected by chemoresistance. It will be equally effective in male and female, but also in frailty individuals that, because of the age, cannot get chemotherapy. The entire solution is based on the use of intravesical installation. So we use catheter and cystoscope. So all techniques or equipment that are of current use in the clinical scenario. And we use gold, that is a biocompatible agent. We expect that this solution will reduce the relapse in more than 600,000 patients, while at the same time we expect to reduce the social cost. That's it. Thank you for your attention.
Thank you very much, Massimo. Now I would like to invite Dr. John Hermans from the project PANCAID. John, the floor is yours.
Thank you, Federica. So my name is John Hermans. I'm a radiologist from the Radboud University Medical Center in Nijmegen, and I have a background both in engineering and in medicine. Several years ago, we started a European conglomerate, which was called PANCAIM, and we aimed to develop multimodal AI in order to improve treatment of pancreatic cancer. As you might know, pancreatic cancer is a very devastating disease with a very poor outcome. And let's say the 5-year overall survival is around 10%, so that's very poor. And even worse is that it has not changed during the last 40 years. For other cancer, that has improved a lot, but not for pancreatic cancer. So for us, that was a good initiative to get started to improve that treatment. So, as you know, let's say when you want to develop AI, you need a good quantity and a good quality of data. And that's one of the most important things also of this project. We collected and connected these data into a nice repository. And when you talk about data, there are a lot of different types of data. So you have clinical data, which have different data formats, the PHIRE or OMOP. You have CT scans, you have MR scans. Those are more or less well defined in DICOM format or NIFTY format. But when you talk about pathology, you have already a lot of different sizes of data representation. That can be TIFFs or JPEGs or even more recently DICOM. And the same is applicable for omics data. Depending on the type of omics, you have also different types of data formats. And we work together with the Hive, a small company, which has a lot of expertise in harmonization of data. And with Hive and PANCAIM partners, we developed a common data model. And this common data model describes different aspects of the patient. So we have personal information regarding age and body weight, for example. We have information about the treatment of the patient regarding resection, chemotherapy, or other treatments. We collect laboratory values, and we have also, of course, the outcome of the patient regarding survival. And all those data are finally being uploaded into this repository. So then the next step is also to connect all the partners to this repository. And all partners, they have a lot of different data, and they need to be facilitated to upload those data. And this transportation and this interconnection of the partners with the repository needs, of course, also a lot of legal support. So we have all kinds of documents for data sharing, data transport, data processing agreements in place. And that has all been facilitated by Collective Minds, which is also a large company with a lot of expertise in this field. So in the end, you will have this repository where you can show that, in this case, Oslo University Hospital is uploading data. And it is shown as a progress regarding the clinical data, the pathology data, and omics data, for example. And then here is a field where you can see the clinical data available. Here is a part where you can have a view of the CT scan, and you have a viewer to analyze the pathology slide. And you also have an overview of the omics data. So then you have all the data in place, but then you need to develop, of course, the AI. And we developed at several points of the patient pathway several types of AI. Sometimes we developed AI to prognosticate whether the patient will get metastasis in the future based on a baseline CT scan. But one of the highlights of this project is that we not only developed AI, but we also implemented AI, and that is a very big step forward. And one of those AI models we developed was about the detection of pancreatic cancer. And we developed that with all partners together with a lot of data. And the final result was that we could share it with some of the clinics where it runs in the background. So here you can see one of my colleagues, David Rutkowski. He's a radiologist in training in the Karolinska Institute, where he's preparing his MDT for pancreatic cancer. And he is using the AI to detect the pancreatic cancer location. And this information is being used in the multidisciplinary team to discuss the case. So that's a very big highlight for this project. So thank you for your attention.
Thank you very much, John. Very interesting. I have already a lot of questions. I would like to go to the next speaker. Dr. Núria Malats will present about PANCAID. Núria, the floor is yours.
Yeah. Thank you very much, Federica, and also hi everybody. Can you see my presentation?
If you can put it in presentation mode, we see it. Yes, perfect.
No?
Thank
Perfect.
you.
Okay.
Perfect, Núria.
So I'm representing the consortium now of PANCAID, that it stands for Pancreatic Cancer Initial Detection via Liquid Biopsy. And actually, early detection of pancreatic cancer is an urgent need. So we can offer the patients with curative treatment, such as a resection. Otherwise, 80% of the patients with pancreatic cancer are diagnosed too late, and they cannot have this curative treatment. So PANCAID aims to provide a minimally invasive blood test using a comprehensive panel of liquid biopsy diagnostics for early detection of pancreatic cancer. The PIs of these projects are Klaus Pantel from UKE, that it is in Hamburg, and Matthias Lohr from Karolinska Institute in Stockholm. The duration of the project is 5 years, 8 countries participate, 18 institutions, and you have the map on your right-hand side with all the institutions participating. The grant, the amount of funding is almost 10 million, and the project started 2.5 years before. So we are now in the middle of the period of this project. What we have done is to biobank the blood samples from patients and individuals at risk for pancreatic cancer. We also have collected detailed data that allow us to understand their level of risk. And nowadays, the experts, the European experts on liquid biopsy analysis on the features, they, for example, ctDNA, exosomes, CTCs, CICs, and circulating proteins and metabolites, they are analyzing both serum and plasma samples. We will have the first results of this analysis later in this month, and we will start combining all these features in order to build a composite multi-marker panel using AI. The project then aims to support the implementation of this blood test into medical practice and to design a protocol for a prospective multi-center studies. So myself, I'm MD, PhD. I'm expert in AI and statistical analysis from the Spanish National Cancer Research Center. I forgot to present myself, to introduce myself. So I'm leading the work package 3 that regards to build this model, integrative liquid biopsy feature models. And here you can see that from the patients we get the sample, the blood sample, the analytes are analyzed, and the features are delivered to us. Well, and then we build this AI model. So there's several steps that we have to follow because this is a complex process. First is data normalization, considering all different types of features, the feature selection that we plan to do that with Boruta finally. And then we enter in the step of the model building, and we plan to use both machine learning and deep learning using different approaches for early fusion, intermediate, and late fusion. For sure, we are exploring the different models that we can build through the classical one, regularized regression, and random forest, that these are more typical, more classical, but also neural networks and GNNs. And for sure, after that, we will select the best model using the standards parameters and estimators, confusion matrix, sensitivity, specificity, and balance accuracy. And finally, very important is an internal and external validation that we plan to do that. So the retrospective part is ongoing, and we will have results very quick on that. And the prospective part is also ongoing by recruiting more patients and individuals, high-risk individuals, with all possible biosamples on that. Thank you very much.
Thank you very much, Núria. Thank you, everyone, for the introduction. I would like now to invite you for a 30-minute panel discussion before we start, or we open the floor to questions from the public. So, as we are talking in this webinar about early cancer diagnosis and early detection challenges, I would like to ask you something about exactly early detection. So why is it so critical, and how are your projects addressing these challenges of early detection in different ways? And what are the biggest barriers for early detection? So perhaps, I don't know, John, do you want to start about that? So the barriers in early detection, why is it so difficult to spot it early?
Federica, of course. Let's say early detection is indeed the holy grail. And maybe it also depends on the type of cancer. For pancreatic cancer, it definitely is. And we know that most patients present very late during the disease. So they present at a stage where the tumor is either locally invasive or already metastasized. So the one and only potential cure could be early detection. That means detection of a tumor in small size, early stage. But then you run into problems because, let's say, most people don't have early cancer in the data. So you need a lot of collaboration with a lot of different clinics before you are able to develop an AI model that is able to detect small cancer types. So that's an issue you have to be aware of.
You mean in terms of prevalence? You mean in the database you have in terms of prevalence of early detection? You have too little to be able to build a model that really helps you, so that you need a larger network of hospital or centers that collect this type of data.
Exactly. So within the PANCAIM, we developed an AI model for detection of pancreatic cancer, but that was across all stages of disease. So from stage 1 till stage 4. And stage 1, that means tumor size is up to 2 centimeters. So that's not really very small, but rather small. So what we're now trying to do is to run that algorithm and see how it works in clinical practice as a research tool. And then you can see, are you able to find any small cancers with this algorithm? And if not, then you need to do the next step, which means you have to refine it. So you have to look for more data sets with smaller cases. Yeah. So that's our next challenge.
Yeah, so what you are hoping is that despite perhaps the low prevalence of early cancer, with the model you are developing, you are still able to detect those as well. And in case not, then you need to enrich your data set.
Yeah, exactly. And there's a good chance we might succeed, because we know from retrospective analysis that depending on the type of data and the numbers of data, that there is an increased detection rate of 20 to 80% by experts on imaging that has been done before the final diagnosis. So it seems to be that there are already signs, rather either in texture changes or duct changes, that might be indicative of the presence of a small tumor. And it is normally being recognized by the experts, but not by the non-experts. And the patient flow is mostly that the patient with early signs will only be seen by the non-experts. After the tumor is detected, they will be referred to an expert center. So that's the major gain, so that you can develop an AI algorithm that would be running in the background of every hospital. And then the AI algorithm would flag this abnormal CT scan. Then the radiologist and his colleagues could look at this scan and say, okay, this might be an early cancer, and then the patient gets its routine workup. So that's the ultimate goal.
There is some hope, I would say.
There is some hope, absolutely.
Very good. So moving perhaps to Núria, how does, for example, these challenges can be faced through blood analysis through your project? How can you maybe complete what John says, or how do you tackle that?
Yeah, sure. Actually, I think that you raised an important point, that is enrichment of the population at risk. So for pancreatic cancer is diagnosed usually with very unspecific symptoms and signs. So then this is one of the reasons that its diagnosis is delayed. The other reason is that it's a super aggressive cancer. So the only way, as John said, is to diagnose pancreatic cancer, but when the symptoms have not appeared, when it's still asymptomatic. And the only way to do that is through screening programs. And this is before the patients come to the clinics. Okay, this is general population. But as you also said, Federica, the frequency of the incidence of pancreatic cancer is not very high. For example, as colorectal cancer, that only with age they can filter the population to a screen. So in pancreatic cancer, what we need is to further enrich this population using other type of information. For example, whether they have new onset diabetes, or whether they are obese smokers and or have a family history of a pancreatic cancer. In those populations, then a screening test like a liquid biopsy is actually the most promising right now. And among those that have positive results in liquid biopsy, they will go to the hospital and they will have offered these radiomics test that John have mentioned before.
Yeah. So when you talk about these blood tests, are you thinking about at home test, or at the GPs, or people who come to the hospital for other examinations? How do you envision that in the clinical patient workflow?
Yeah, the patients that will enter in screening programs, the same as colorectal cancer with occult blood in a stool. So they will have actually done these blood tests using an array with the features that we finally have selected through all the pipeline that I mentioned to you. So these will be labs that will perform these tests, and the patients will then be informed whether they should go for a further checking or not.
Yeah. Okay, very interesting. Massimo, do you want to add something with respect to early detection and therapy from bladder cancer?
Yes, so as for other kind of cancer, in the case of bladder, the first symptoms to make a diagnosis, the first symptom is the presence of blood in the urine, then followed by other investigations such as urinary cytology, meaning we look for cancer cells in the urine. And so this makes plus other ultrasound imaging or MRI that confirm the presence of the cancer. In case of PHIRE, we aim to do early detection of very small lesions. Okay, so once there is a diagnosis of bladder cancer, we aim to detect that very small lesion that will, if not treated earlier, they will give rise to the relapse and potentially to progression. So it means that if there is a progression, the cancer is spreading through the bladder and goes into the other organs. Then the main the guidelines suggest to completely remove the bladder to avoid spreading and metastasis. So with PHIRE, we aim to do early detection of lesion that will probably give rise to relapse and eventually to progression.
Yeah. So what you're saying is that in any case, the detection will be done through presence of fluid in the urine. And once this is done, what you aim at is really detecting very small lesion, millimetric size that would not be otherwise visible. And therefore, you kind of are able to target the therapy very, very soon before it is developed and perhaps become metastatic.
Yes, so when there is already a diagnosis of bladder cancer, so the patient that goes into the surgery room, we give the chance to remove the lesion that otherwise the surgeon will not detect.
Yeah.
And so hopefully to stop the disease progression and relapse.
Yeah. Very good. So I think now what I learned from you three all is that AI plays an important role. So we talk about data, we talk about enrichment, but exactly what can AI see that clinician cannot see? What is the early detection and pathological or radiological diagnosis or sign, Jones, that AI helps you to identify that the eyes of radiologists cannot see? Do you know already something about this black box, or is it still a mystery to you as well?
It will always remain partly a black box, at least also to the current standards. But we use, of course, also these saliency maps, for example, that you highlight the voxels and the region where the AI sees something. But let's say one of my colleagues, he's already been doing AI research for 25, 30 years, and developed AI for tuberculosis in third world countries. And he said, what you can't see with a human eye, you won't also see with AI. And I still believe it, but let's say what AI is able to see, it is more able to see small changes.
Okay.
So, but even when you look as an expert afterwards, when an AI assigns something as a positive finding, you will see some changes. So I'm not really sure if AI will see something that the eye won't see. I think the most powerful contribution of AI is the power of computation of multimodal, multiple data points into a single diagnosis. And the human brain is not able to use that enormous quantity of data to make up a diagnosis. I think that's the most powerful impact of AI.
Yeah, so also knowledge representation, the connection with the different type of data, small changes, and that is where the difference will be. Núria, do you have anything to add to that?
Specifically in the liquid biopsy field, each of these type of features that I mentioned, CTCs, ctDNA, each of them actually are represented by thousands of characteristics that AI help us to reduce the dimensionality, to understand better their correlation between different layers and within and between layers, and also how they interact. So finally, we can end up with an algorithm that is a robust algorithm that can be implemented into the clinics or into the prevention in such a case. No. So this is how AI helps us in managing, reducing the dimensionality, and finally resulting in this very robust algorithm.
So identifying the most important or relevant parameter for the cancer detection. I understand you said we will be, that's what help us to develop a robust AI algorithm. How do you test robustness, trustworthiness?
Very good question, Federica. Yeah, sure. No, there's different steps in the process. That is, first we need to see whether it holds internally if we change the parameters. This is the internal validation. We also need an external to assess, to apply it in an external database, completely independent database, to see also whether the algorithm holds its performance. And we need to assess the calibration, and finally, very important, is the clinical utility, okay, of this, because it may be a fantastic, very robust, but with a small clinical utility.
Yeah. Adoption stays a very important point. We see a lot of AI developed with very low adoption, which is another topic, probably not the one of this webinar. Massimo, do you want to add something? How do you tackle the AI challenge in your project? Do you leverage it? How do you use it?
In the case of bladder cancer, and we tackle the AI models in a different way, because in our case, we want to apply AI-based algorithm to the thermal therapy, meaning that we want to know exactly how the condition, how much power to provide to the gold nanorods for how long, in order to increase the temperature and do thermal ablation. So we want to know all these parameters, because all these parameters will be different for every patient, because it depends on the dimension of the lesion,
on the location,
of the location, and in particular also on the volume. How much urine or how much liquid is present inside the bladder of that patient in that moment, because we know that the liquid will dissipate the heat that is generated by the nanorods. So we want to, in a real time and in a personalized way, we want to calculate all the conditions, the setup to induce thermal ablation that is efficacious, and try to avoid to make a hole in the bladder wall. So we want to integrate all the different parameters that will be different for every person in a model to carry out the best thermal therapy possible for that kind of patient.
Right. And if I move then to a next question, so linked to AI. So the food of AI is data. John started showing this very nice graph with all the data, different type of data, the ETL, so extract, transform, and load this data in the right format to be digested by a platform. Now, we talked about prevalence of disease. In some cases, this is very low, so difficult to get enough data. What do you think about building large and diverse data set? How difficult it is for your project? And for example, could the synthetic data play a role there? I don't know, John, again, do you want to start, or Núria, one of the two? Perhaps this time we start with the ladies. Núria, do you want to comment on that?
Yeah, thank you, Federica. This is a very important and nowadays very hot topic, no? Synthetic data. So the problems with retrospective data, pulling different data from different centers, is the heterogeneity of the data, the lack of data, lack of standardization. So despite we have some methods in order to impute, to normalize, and etc., but it never ends up with a very nice data. So prospective studies are very costly, and actually the duration goes beyond this kind of project. So we started also generating data, omics data, and also pathological images in order to increase the sample size, at least for the training, okay? We think that the validation needs real data to validate our models. But for training, we think this is gonna help us a lot. So, Actually, I have to say that for pancreatic cancer, we have generated pathological images that the best pathologists in the world that I
Could
have
not
sent the
distinguish.
images, they cannot distinguish, the real from the synthetic ones. So spectacular, yeah. So that
Okay,
field,
so you really see a role for that.
yeah, for sure. I'm very optimistic on that, yeah.
Very good. John?
Yeah, it's a really interesting subject. On the other hand, I would say there have already been acquired a lot of data also in pancreatic cancer research. So I think it is absolutely worthwhile to connect all those people that already have data in their possession. Because I think
like
the real
creating a large databases like worldwide, which is freely available or almost for research purposes. AI for good.
Yeah, so I think for also maybe the EU or EIC, it is a good starting point to have a consortium building on collection of data, curating data, and then you can use it for defining a task-specific oriented AI.
Yes.
And if then you don't have enough data, you can always try to develop synthetic data. And that's always a possibility. But I think you should use already existing data to start with. And that takes a lot of effort. On the other hand, doing so, you also connect other experts in the field. And thinking on beforehand, which is the most relevant question to tackle, you need to have some kind of scientific advisory board with all people involved from the clinics, from health technology assessment, from AI experts, and that are directing towards one goal and tackle one problem at a time and divide those problems across the world. Just like HIV in the time, let's say, many people were involved tackling the issue, and it was resolved very quickly because everybody came together and they distributed different tasks across different domains. It was a unique process which sped up everything unprecedented. So I would say that you could apply it and you should apply it to any medical problem.
Absolutely. And I think I just connected to some challenges that we have brought out as AC. So there is a call out that will close on the 29th, which is exactly about partially also building what you are asking. So at the European level for the moment, not worldwide, but building a mix of real clinical data and generation of synthetic data for cancer, any type of cancer, leveraging perhaps also screening databases and so on, because I really believe in that. I think at the same time, we all know that data are the new currency. So not everybody is as open-minded as you are, and that's a barrier we really see. So there must be perhaps a new legislation on the HDS, so the European Health Data Space with secondary use of data might be a help, although this will be not before, I think, 8-9 years. But that's a way Europe is trying to leverage what you are saying. So thank you for that. Massimo, do you want to add anything? How is it in PHIRE? Do you use synthetic data? Do you have enough clinical data for AI training?
We are using data from literature, but mainly just 2 weeks ago, there was here in my lab a colleague, a partner from PHIRE. We are testing in vivo in preclinical models, and we also use ex vivo organs such as swine bladder. Ex vivo swine bladder. So yeah, we are trying to calibrate and to validate the model on so far ex vivo or in vivo in preclinical models. Not yet on, because before going to the patients, we need to, I mean, there are other steps we taken.
Yeah, I think but for you.
But for us, what was important, we use tissues from patients to identify the marker of bladder cancer, because our goal now are targeted against the tumor marker.
Yes.
So we identify the marker by using tissues from patients.
Right.
That is what we used.
Yeah, and this is because you are at the earlier stage of development and technology readiness level, I think.
Yeah, although we spent already 7 years, but yes,
Yeah,
we
but
have. No,
big discoveries, big discovery. If you think back
Take
at
time.
the Apple Watch took 20 years. I
Yeah,
mean, it's
that's
not
true.
comparable, but that's breakthrough technologies they are not developed in 3 years, and that's also where perhaps they see struggle sometimes, because with the instrument we have, the timeline of the development of breakthrough medical devices does not necessarily follow the timeline we have of 7-8 years, nor the timeline of the investors who expect return on investment in 4 years or even short or 7 years, like they would expect in green or other technologies. That's not the case for healthcare.
If I can add, I think that this applies to PHIRE, PANCAID, PANCAIM, but all projects in general, not only medical devices, but also for the artificial intelligence. I believe that they will need some certification. I mean, the software, the model will need some certification. This will take time and a lot of money. So you're absolutely correct.
Yeah. And if I connect back then to the AC, which I represent, so one of our mantra of the research we fund is we want to bring research to the market. And that's why we have 3 instruments: the Pathfinder, the one you have, which is very low TRL. Then you have the Transition, which is in between. So once you prove, the concept that it works, you start building a business case, you try to do preclinical studies and so on. And the Accelerator, which is for SME, so to scale up your idea. And as I said, our mantra is bringing research to the market. So if I can go back to the 3 projects, what do you think are the steps needed to move your research to real world clinical implementation? Do you want to start, Massimo, maybe?
Personally, I mean, as a coordinator of the project, I would like to have, because I'm a scientist, I'm not an investor, I'm not a businessman. So many of these steps about regulatory pathways, certification are quite new to me. So probably from European Union or from ESC, it would be very nice to have some support about regulatory frameworks, some practical help, no?
Yeah.
I don't know, some project manager dedicated to regulatory frameworks, to certification. So otherwise, I do not by myself, but I need to ask support to external services
for experts.
To pay. So probably it would be nice to have someone from ESC, that could guide us in these pathways that are new to researchers.
Yeah. So I take the opportunity.
And I think that this is even more important for AI. That is a new field, a new topic that what I understood. There is nothing sure in this field. It is novel, so they are built on this.
So I think to that I take the opportunity to share that we are organizing an event tomorrow with the NoboCap, which is the consortium of the notified bodies in Europe. I think you can still register to participate online. This I say it also to the attendant of the meeting, where we are exactly discussing this topic, the definition of breakthrough technologies, how can help the researcher and the innovators to bring their research to the market. So very welcome to join. John, do you want to add anything to that?
Yeah, I think implementation of an AI model in real routine clinical practice is one of the most difficult steps. And as you look at the complexity doing so, you need also large expertise with in different domains. And I think it is also unavoidable to connect and to collaborate with industry, because let's say, as a university hospital, you are not able to implement an AI in the clinic. Also within PANCAIM, we collaborated with Siemens, and they have, for example, a very useful platform, which is called TeamPlay, and you can upload your AI algorithms, and then it is made available in thousands of clinics worldwide. Yeah. And such a platform that has been regulated, is safe, and so on. So you need that kind of collaboration.
Yeah.
I think strength of university could be also in a different role. Let's say we also play that now within the PANCAIM project, that we are setting up a benchmark. We not only develop AI, but we are setting up a benchmark which is used for the trustworthiness of AI. It's only one of the parts of the trustworthiness, but it's very useful. So you design a system where you can set up a challenge where AI teams worldwide and clinicians worldwide can do a referencing, and you check if the AI is as good as the clinician.
Yeah,
and that outcome that can be used. Yeah, that is useful. You have a benchmark, and this benchmark can be used worldwide to improve on AI. So you have a dynamic AI model. It is not fixed. It will change during use.
Yes.
And so that could be a very good contribution from the university or any team that is developing AI. But it's really complex, this last step of implementation.
That's why adoption is hindered a bit. So we have like 1 minute before I would like to start the Q&A. Núria, do you want to have just a one-shot message to add on this subject? If you can unmute yourself.
Sure. AI is a needed tool for all of these projects that are integrating or analyzing images in order to finally get real implementary devices into the clinic. So we do not have to fear AI. We have to enter AI to teach AI to our students and to train them to use the AI in the best way in order to help all our projects. Yeah.
Absolutely. While trying to avoid de-skilling, you know, that for example, the radiologist doesn't become lazy because AI will find it for him or for her. Great. Thank you very much. So I would like now to open the floor for questions from the audience. I see that there are already some questions in the Q&A chat. So please ask your question there, and I will be very pleased to pass it to our distinguished speakers. I will ask the first question, which has been asked by Massimo. So to John, it's for you. About early detection, any hints from preclinical models of PCA?
Could you repeat the question, Federica?
So in terms of early detection of cancer, do you have any information or any hints from about preclinical models of PCA?
For relating to imaging, we do not have any models available.
No, not yet.
But we have to start from scratch on.
Okay. I was thinking maybe if there is some preclinical model of pancreatic cancer, maybe you can follow the steps of the development. So if you can learn something from the preclinical model to see how the disease evolves over time or during some kind of experimental treatment.
Yeah, but Massimo, at the radiological level, mice are so different than humans. So yeah, it's difficult. Maybe at the molecular level, this is more feasible. Okay, and to see how the molecules, liquid biopsy features, they can evolve over the evolution. Yeah.
If they change based on the treatment, and then you can model this change. Or some genetic mutation that are different in different preclinical models.
Yeah. No, in liquid biopsy there are preclinical models.
Okay, then we have another question from Ernest to all of you. So feel free to take the lead. So the question is if you are open to the use of agentic AI. So agentic AI, which use information to do reasoning and taking autonomous decisions. So let's say saying you are developing several models, several AI models, run the data through all of them and ask agentic AI to interpret and for example, take decision or give recommendation to the human, to the medical doctor, for example, to lower the workload or to augment their, let's say, capability to detect cancer, for example. Anyone?
Yeah, I think that's a real important question. In the end, we will use agentic AI, but currently we're not that far yet. So we need first to develop a lot of good working tools. That will take some time. But in the end, let's say this kind of agentic AI is inevitable. The power of such AI is enormous, and everybody knows it, but it still needs to be developed.
Absolutely.
Yeah, fully agree. Yeah.
There was also a very interesting talk at the Italian Tech Week in Turin a few weeks back, exactly about the same topic. I think the conclusion was this will come. It's just a question, they gave the example that the ChatGPT was just started, 2 years ago or 3 years ago, I don't recall, and that nobody was expecting the impact that we have today. So things are going to be very fast, and we better be ready, I guess. Elisabetta also has a question for Massimo. Massimo, this time is for you. Can gold nanorods be used for detection other than cancer lesion? Like, can they work also on organs other than tubular ones? 2 questions in one.
Okay, so thanks, Elisabetta. So the gold nanos that we use are targeted. And to the best of my knowledge, all the nanoparticles in general that are used to detect or to treat diseases are targeted. So there are colleagues of mine that are using targeted nanoparticles to detect small brain tumor or against the glioblastoma. So to me, all the nanoparticles must be targeted to reach the site or when the organ of interest. Otherwise, they stuck mainly in the liver. About non-neoplastic diseases, for sure, they can be used. Depending on the disease that you want to tackle. For example, to me, you could use nanoparticles to recognize collagen, for example. And in this case, if you apply targeted nanorods into the blood system, maybe against collagen, maybe you can detect collagen in the atherosclerotic plaque, in the active atherosclerotic plaque, that are those that are putting the patient in dangerous. So yeah, you can use for neoplastic and non-neoplastic diseases if you're able to identify the marker of the disease.
Yes. Okay, thank you very much. Then we go for one last question, or maybe last 2. So this question is, I guess for Núria, can blood samples provide sufficient sensitivity for the early detection of pancreatic cancer, or is there a need to look at organ-specific fluids like pancreatic juice, for example?
Well, actually, the idea of integrating different features aims to increase sensitivity and specificity for this tool. And in ending up with a non-invasive device to detect pancreatic cancer. If we plan to apply this algorithm, this array for screening purposes, we cannot think of using pancreatic juice to increase sensitivity and specificity. So this is our goal. We will see to which level of performance it achieves, but then by combining other kind of tools like images, imaging analysis, etc., then we will think that we could decrease a lot the false negative and positive cases.
Yes. Okay, then the last question still for you, Núria. Any challenges encountered in reaching out to patients for recruitment purposes? And did you have any communication-related obstacles? Any suggestion for a newly project like SHIELD, which is just started? They are developing a blood test to detect pancreatic cancer as well.
So thank you for all these questions. Okay, well, in Spain, at least, recruitment of patients and subjects, healthy subjects, is not does not represent a real hurdle, because here people regarding these healthy topics, they are quite generous and they participate. However, pancreatic cancer poses several challenges here, and patients, mainly for patients, most of them they are very sick when they are diagnosed. So to have them participating, not because their willingness, but their possibility to provide samples, a large amount of blood or different types of samples and information. This is difficult. The way we do that is we address that is with monitors, trained monitors, that they are very motivated and they can translate these motivations to the patients and the families. Families are also very important here. Absolutely. So maybe this may work also in your setting.
Thank you very much. So I would like, we are going towards the end of this webinar, but I would like to ask one last question to all of you before we close. And the question is, how does the future looks like? Shall we envision a future where early cancer detection will be a reality or not in the next 5 to 10 years? Just few words, yes, no, and why? Who wants to start?
Should
Núria.
I start?
Yes.
Yeah. Yes, a big yes. And
Very
we
good.
need. I'm working on pancreatic cancer for the last 25, 30 years, and this is the momentum. This is a golden age. There's many groups working on pancreatic cancer nowadays, and actually from different angles. And I think that we will be able finally to get an early detection of this disease when it's still asymptomatic, and identify super small lesions that will be susceptible to get cured. Yeah.
Thank you. That gave us hope. Massimo?
Yeah. If okay, so I fully agree with Núria. The answer is a big, great yes. Also because in this moment we have technological possibilities that we never had before. So including AI models. So I'm very, because in the last, I don't know, 20 years, we generated a lot of data, a lot of information that we were not able to integrate together. So now we have technological implementation and the way to use all this information that is given by artificial intelligence. So
Very
the answer is yes.
John, I expect a yes from you.
Absolutely. Yeah, there's no doubt about it. It's definitely a yes. I think also, let's say these new developments, they show us that you need to think and work in a different way. Doing research is changing. So before you start a project, get a large group of people together, a large group of experts. Try to collaborate. Collaborate is necessary, but it's also fun. And it will, in the end, it will improve your clinical impact. I think that's what the main goal of research should be. Try to achieve clinical impact, not doing research for the research, unless you're doing really fundamental research. But when you are involved in clinical research, try to get a very clear, defined clinical impact project.
That's exactly what we want also at the AEC. So today we have seen really how health technology and AI are reshaping early detection of cancer. I hear from you that there is hope with approaches ranging from imaging to blood to molecular data. And I think what I retain the most among many other things is what John says a couple of times is that collaboration across discipline is really necessary to bring this solution, personalized solution to the patient. And with this, I would like to thank you very much for this pleasant discussion. I learned a lot, which is great for me. I hope the audience also enjoyed it. And I wish a wonderful afternoon to every one of you. Thank you very much for attending and for being here.